Hello class, this is Dr. Ogilvy. I'm going to review some of the questions in Module 1 Part B quiz. Before I get to the quiz, I want to point out what we have shared with you in the syllabus about the quizzes and exams, and particularly for Part B. And here on page 12, we have shared with you that the 20 to 30 questions in Part B of the module quizzes and on the midterm, and in the case of the final there'll be 3 parts, or it'll be Part C, are virtual slides, and most of the time these will be unknown specimens that you have not had seen prior to the exam, so that we can test whether you are just relying on rote memory of a certain specimen, or whether you are using a reasoning process and thinking about the criteria or the diagnostic features of a cell, a tissue, an organ, or part of an organ. So in answer to these questions, you can navigate and zoom in and out. So I just wanted to make sure that you're aware of this and you can read that for yourself. So now, unlike Webmic, that I trust most of you are taking seriously and using, where in a Webmic specimen, if you're studying a certain tissue and we navigate, let's say you're going to study skin, and with skin you get certain predetermined magnifications, and these are nested images. You get a low power image, and then there are high power snapshots throughout that particular specimen. And if you remove the screen, you can see that it is a snapshot. An individual specimen you cannot zoom in on. And so that's much different than with Lucido, where you by now hopefully have set up Lucido and it's working well for you. So in addition to studying Webmic, where you're learning ABCs of practical histology and trying to match what you learn in lecture with the Webmic information you learn from those specimens, we in each of the modules, and it was true of Module 1 and Module 2 and 3 and 4 and 5, there is a folder that has Module 1 Lucido Lab specimen videos. Now these videos are taken of the specimens in the Lucido collection for labs, and I'm going to bring that over and just show you that, as well as the ones for Module 2 that you're working on now. So when you use your cloud access for the Lucido Lab specimens, you have a collection of specimens for each of the modules. And for Module 1, you had skin, trachea, 2 actual samples of trachea, urinary bladder, esophagus, colon, salivary gland, and you had actually a mouse specimen that we did partial annotations on, and this is a specimen of brown adipose tissue that we invited you to study. And you see, if you match this with what you learned in lecture, there are the unilocular adipocytes, and this is white adipose tissue. And here are the multilocular adipocytes. Each one of these round blue structures are nuclei of the multilocular adipocytes that have tiny lipid vacuoles as well as lots of mitochondria. So if you take each of these specimens and we give minimal annotations. In this particular one, we have given, and you can zoom in on these and study and practice thinking about the criteria that differentiates covering epithelium, which here is stratified squamous keratinized. See all the keratin here, and then the live cells here, and then the connective tissue where you have dense irregular connective tissue down here, and then just underneath the epithelium, you have loose connective tissue. You see how fine the fibers are and the fibroblasts, etc. So what I'm trying to do here is to get you excited about taking advantage of each one of these specimens. So Module 2 is what you're studying now, so you have an example of hyaline cartilage, decalcified bone, ground bone, developing bone, and so on and so forth. And for each one of these, we will have some minimal annotations. The idea is not to give you enough information so you can get in here and study them and relate that to lecture and lab. If you put it all together, histology is about size, shape, relationship, and function, and the appearance after we have stained with hematoxin, eosin, or other specimens. Okay, so now I'm going to go back to the course website, and I'm going to show you some of the resources for exploration outside of Webmic and Lucido and the lectures. Okay, so here are outside resources. And we know that histology is not it's not that easy. When you really get the hang of it and you are looking at in the lecture and the lab and you are making notes and you say, okay, this is a mast cell. So where do I find mast cells? And what do they look like? Well, they have granules in them. And what is the shape of the nucleus? In this case, it's round. And where they're found, whether they're close by blood vessels, etc. What did they produce? Well, serotonin is one, and histamine is another. So you see, when you think about mast cells like that, and you begin to put that down on a 3x5 card, or however you want to do it, to do the neuromuscular activity that will help you remember this, then you can go to the outside resources. And in the case of virtual slide exploration, we actually give you a PDF that I'm not going to go through, but we've tried to give you an idea of how to go about approaching a virtual slide, an unknown slide. So that's one resource there. And then a pathologist created these three to maybe no more than 10-minute walkthrough, and there's page after page after page. You could, I don't know that you can do a search, but you can just thumb through those and find. If you're studying pancreas, you might want to watch this one. It doesn't take that long, but the more you combine these outside resources with what you're given in the course website with Webmic and Lucido and the lectures, the better chance you have of doing well in the course. And here is a site that is absolutely marvelous. For example, I think we're studying muscle tissue. So this is, you click on the slide box, and let's say here is muscle tissue. And all of these are virtual slides or whole slides. So here's skeletal muscle. And the way that the professors up at the University of Minnesota put this together, if you just click on the cross-section, it takes you right to a cross-section. Then you can zoom in and study this, or you can just click on skeletal muscle cells and it takes you right there, and it gives you some basic information, and as well as the longitudinal section. And if you click on like this, it'll bring in the myofibrils. Now you see the banding, the A bands and the I bands and the Z disc. You can't go to, well, over here is a pretty good example. You can almost see the Z lines here. Okay, so I'm going to go back here, let's see, and also histology on the internet. Now, I'm not going to go through these, but you might want to at least visit these once to see if they fit into something that will help you. And then we have recommended texts here, and those texts are not required. And we also then have PowerPoint lectures that you can zoom in on and download and then open them. And I'm not sure how fast this will open. I'll pause here. Yes, this is an example of Dr. Bereford. He is deceased now. I visited him at the University of West Virginia years ago. He spent hours upon hours and made all these things freely available over the internet. For example, epidermis. You're studying in Webmic and Lucido in the lecture, but if you come out here, you can see he's diagrammed this beautifully for you. Not saying you have to do this, but the more you can take advantage of different exposures, different ways of exposing yourself to the same information, the better chance you have of retaining the information. Okay, so we also have some out, and I need to try to put some more here, but these are places on the web that you might want to watch that are very helpful and expand. And then, of course, we have color deficiency and text anxiety help, etc. So I'm going to pause this now while I get the quiz ready, and we'll spend a few minutes going through some of the questions. And maybe with everything that I've presented so far, you can begin to see how to put your thinking together and organize your thinking when you approach a virtual slide in Part B of the quizzes, midterm, and the final exam. Okay, I'm back in Module 1, Quiz Part B, and this is the first question. Of course, when we deliver the test to you, your order of numbers of the questions are changed with each new login. But this is question 1, and we ask you to provide the dominant cell type present in the tissue samples that were outlined by dashed lines in the virtual slide. And the best answers would have been fibroblast or fibroblasts, singular, fibrocyte or fibrocytes, or we even accepted tendon cells, which is not exactly correct, but it's related because the tendon cells are modified fibroblasts. Now the specimen that when you clicked to load the specimen, this is what you were presented with. And so if we enlarge this, the pattern we see here are eosinophilic structures that are like wisp, and actually they're different profiles of fairly thin collagen fibers and a lot of space, which would be ground substance that was washed out when the tissue specimen was prepared. And then you see these nuclei, and some are elongated, and if we enlarge just a little bit that one, you can see it's totally out of focus, but you can see it's longer than it is wide. And this suggests that it would be a fibroblast. If you want to go back to the original magnification we presented, just click on the annotation and then click Go To. If we look at the other region, we even see the nuclei better and the collagen fibers, but they're not very thick, and so the cells that produce those pink structures, the collagen, are fibrocytes. Now, when you look at the response, only 20% of the class got this correct, and we can make a tally of all of the different incorrect answers that were put in. You can see here this person answered epithelium, but we asked cell type. This person answered connective tissue, but we asked cell type, and so on and so forth. This person said mesothelial cells. Well, those are epithelial cells, and we haven't even really focused on those. And simple is certainly not an accepted term for any cell. Simple really needs to be tied to some type of epithelium. Simple squamous, simple cuboidal, simple columnar, and of course, but anyway, when you look at all these, and of course adipose cells, those were not adipocytes. And if we look at this again, those are not adipocytes, but these are adipocytes here. You see a very empty space surrounded by a limiting membrane, which is a little connective tissue in the cell membrane. And then occasionally you can see a nucleus here, and if we go higher down at lower magnification, here's another adipocyte, there's the nucleus, and that's a unilocular adipocytes. So this reminds me to remind every one of you that when we ask a question, make sure that you understand the question. Now I'm going to search for another question that will illustrate another point. I'm not going to go through every question, just enough to give you some hints and some direction of how to think about approaching these unknown specimens. Okay, this is question 4, and you are asked to identify the tissue within the blue rectangle in the virtual slide specimen. And you were given a multiple choice question. You actually have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 possibilities. And 55% of the class chose the correct answer, simple columnar epithelium. 30% chose simple cuboidal epithelium, and 5% chose simple squamous epithelium. Let's look at the specimen. Here's the specimen. When you look at this specimen and say enlarge it a little bit, you can see the cells are taller than they are wide. That immediately says it has to be simple columnar epithelium. Even though the nuclei are somewhat rounded, you have to combine two bits of information, the height of the cell and the width of the cell in its relationship to one another, and the nuclei. And the fact that there's only one layer of nuclei indicates it's simple. Of course, out here you can see the boundary line here as I'm using my cursor and showing you this is where the basement membrane would be, and out here would be connective tissue and blood vessels. Now I can look down and see probably will not find any cuboidal epithelium here because this is in the highlights of the kidney, and I'd have to look up in here. Maybe we can find some here. Okay, this is more like simple cuboidal. You see the cells are about as high as they are wide right in here. Now I'll enlarge that just a little bit more. See, simple cuboidal epithelium. All right, I'm going to go to another question. Okay, this is question 15 in part B of Module 1 quiz. 2 large cells are enclosed within the dotted line. Examine the nuclei, which are yellow arrowheads, and the nucleoli and the 2 cells indicated by the yellow arrows. And then you were asked to select the true statement about these cells. Were they inactive at the time of fixation? Were they synthesizing proteins? Were they synthesizing steroids? Was the DNA of the nuclei of these cells, was it condensed? And were the cells in the metaphase of mitosis? 30% of the class got the correct answer. 20% said they were inactive at the time of fixation, and the DNA of these cells is condensed, 35%. So let's just look at the specimen. All right, these are the cells. Here are the yellow arrows, and they're pointing to the nuclei, to the outside of the nucleus. And so if we enlarge those just a little bit, you can press Z at any time and use the marquee and bring them up and see a little bit larger. And I think that we explained this in how to approach in a video that we gave you and how to interact with these virtual slide specimens. So here, this dark round structure is the nucleolus, and then the nucleus is here, and the nucleus has extended, not condensed DNA, and of course the wisp of blue out in the cytoplasm would be ribosomes on rough endoplasmic reticulum. Now here's where you had to combine what you learned in the cell lecture with looking at this specimen. You had to think about what is the role of the nucleolus, what is the role of the nucleus, what is happening when the chromatin is distended in the nucleus, and that combined the extension of chromatin as well as the nucleolus suggests that this cell was synthesizing proteins. And this is a neuron, and they synthesize proteins continuously as long as they are alive. Okay, the next question is question 17. And The question stated, the structure encircled is a blood vessel in which you can see a neutrophil and a lymphocyte. The correct name for the tissue making up the wall of this vessel is, and you were supposed to type that in. And these are some of the possible correct answers. Now we were very lenient in this. In fact, this answer here, I doubt if anybody would have put this, and that would not. I mean, it's true, but there isn't any such thing as non-keratinized simple squamous epithelium. All keratinized epithelium have to be stratified squamous. But all the rest of the answers are acceptable, and 65% of the class answered at least one of these answers. And I'm going to show you the specimen, and then we're going to see the answers that were submitted that were incorrect. So here again, the structure encircled is a blood vessel. This blood vessel here, and in which you can see a neutrophil and a lymphocyte. So there's the neutrophil here. It has a kind of a horseshoe-shaped nucleus, and there's the lymphocyte, which is a round, dense nucleus. And then you have the lining of this vessel, which is endothelium, or simple squamous epithelium. And that was the correct answer to this question. That is the best correct answer, either endothelium or simple squamous epithelium. Endothelium is the term given to the lining of blood vessels, and simple squamous epithelium is the type of tissue that endothelium actually is. So if we look at the answers that were typed in, someone typed in collagen, and we asked the lining, and certainly it's not collagen. In fact, the question said the correct name for the tissue. Collagen is not a tissue; it is a structure. Adipose is a tissue, but it's totally incorrect. Basal lamina is not a tissue. Lumen's not a tissue. Dense irregular connective tissue is a tissue, but that's incorrect. Adipose tissue and then connective tissue, incorrect. Hopefully that will stimulate your thinking to really begin to diagnose and differentiate. And you have to know something. You have to know the possibilities to narrow them down. And this is what happens as a future healthcare provider, whether you're a medical doctor or a dentist or a physician's assistant, a nurse practitioner, or whatever. You have to be able to have enough knowledge to understand the possibilities and then narrow them down. That is differential diagnosis. Okay, so I'm going to go to one more question. Okay, here's question 20. It states the black substance indicated by the arrows in this virtual slide specimen is foreign to the body and is contained within a specific cell type. What is the cell type? So the possibilities were macrophage, macrophages, histiocyte, histiocytes, macrophage cell, and macrophage cells. Those are the possible correct answers we could think of that you might type in. So let's look at the specimen. Here it is. And we're pointing out the black substances. And when we say black substance, and we also give the information foreign to the body, then you need to begin to go back and think, okay, what is foreign to the body? What cell interacts with foreign substances? And if you remember the lecture on connective tissue, and then some of the Webmic and BioLucid specimens, and if you went out on the web and learned about macrophages more and reinforced, you should, with all these clues, come up with a cell that eats, the large macrophage, the large eating cell, which actually is derived from circulating monocytes that come out in the tissue, and then they become active, and they can engulf by phagocytosis this foreign substance, which are probably carbon particles, or they could be smoke particles that are accumulated in these cells as a result in somebody that's smoking. Okay, so I want to show you the possible answers that were submitted here. Remember now we asked for the cell type. So one person put blood vessels. We did not ask you blood vessels. Lymphocytes, they do not ingest foreign substance. This was an epithelium and all these. Now monocytes is really incorrect because the proper name for cells once they're active and ingesting foreign substance would be macrophages. Okay, so now you can see where some of the thinking went incorrect. I hope this has been helpful, and I really hope that everybody in the class watches and listens to this video to the very end. It's just a little over 25 minutes long.